Okay, so here we go with continuations of cells and organs of the immune system, and then we are going to start with more specifics on the innate immune system. So we had stopped on this slide before, and we briefly talked about the lymphocytes. Right? Remember, we have T cells and B cells and innate lymphoid cells, and that includes the natural killer cells. Right? So we briefly went over a little bit of details on those cells. So we mentioned that the T cells, you have two types. You have the T helper cells and you have the T cytotoxic cells. So your T helper cells, they have a specific marker on their surface. Those markers, we call them cluster of differentiation. Actually, most of the cells have different clusters of differentiation. And we identify the cells based on which cluster of differentiation or which combination of cluster differentiations they have on their surface. Right. So for the T helper cell, they are going to have cluster of differentiation 4. They're going to be CD4 positive, and they are also going to be CD3 positive. Okay. So your T helper cells, there are many kinds of helpers. And the development of those helper cells really will depend on what type of pathogen they need to fight. And we're going to go into more details on that a bit more today. Okay? So T helper cells, they are going to recognize antigen that are presented to them in a special molecule that is called major histocompatibility complex type 2. Okay. The molecule on the T helper cell that recognizes MHC2 is actually the T cell receptor. Right? So the T cell receptor recognizes this molecule, this protein molecule that is present on MHC class 2. Okay. Now, how are you going to remember that? Well, here we are going to say T helper cells have CD4. They recognize MHC class Q. 4 times Q is 8. Okay, that was supposed to be a simple question. All right. So that's your T helper cell. Now, looking here at your cytotoxic T cells, they have CD8 on their surface. They also have CD3, okay? And they are going to recognize a piece of protein that is presented to them on a molecule that is called MHC1, okay? So MHC1 molecule on presenting cells are going to interact with that CD8 part and is going to interact with the T cell receptor. So these cells also have T cell receptors, okay? And then they're going to get activated. After they see the antigen that is presented on MHC1, then the naive cytotoxic T cell will become some effector T cell. So before they see antigen, they're all naive. After they see the antigen, they become effector cells. Okay? And that is a common feature of all of the lymphocytes. Okay? Now, look what happens here. CD8 times 1 is 8. So you just multiply those CDs by the MHCs and you should get 8. So that will check that you know which T cell goes with what MHC. Okay? All right, so here, that was a brief overview of the T cells. And then we'll look here at your B cells. Okay? B cells, of course, before they see the antigen, they are naive. And then they see the antigen and they are going to also become more specialized. Okay? Basically, there are two types of B cells. There is the type 1 B cell, so B1 cell, that is mainly present in the immunity of the intestines and some mucosal tissue. So mainly in the intestines. Okay? The B1 cell, what they're going to do, they're mainly going to produce a type of antibodies that is called IgM. Okay? So this is a bulky antibody that has 5 antibody molecules that are connected by joining things. We're going to see that when we get to the antibodies. Okay? So those are very, very important for mucosal immunity. And this type of B cell is also important for production of antibodies against those blood types, the AB antigens, right? So AB group antigen for the blood types. And I'm sure everybody here has heard about different blood types and what kind of antibodies you produce, right? In case you are A type, which kind of antibody do you produce? Anti-B, right? Because if you have A, that is self, you're going to produce antibody against something you don't have, so that's anti-B. If you are B type blood, what kind of antibody you're going to produce? Anti-A. If you are AB, what antibody you're going to produce? None, right? And then if you're type O, you produce both A and B, right? Remember, we also have the Rh factor. That's a different story. We'll get there sometime. Okay. So B1 B cells are mainly very important for mucosal immunity, and they're going to produce IgM. Okay. Then you have the B2 type of cells. That is the type that you see in the circulation, that you see going around in the blood. That is the type that is going to sit on your areas in the lymph node, in the B zones, in the lymph node, and they're going to be sitting there waiting to get activated in case you find a specific pathogen that they have developed against. Right? So that's the type that you're going to see in the circulation. You're going to see in the blood, you're going to see traveling through the lymph, right? So that's the kind that moves around everywhere. Okay? Before they get to see antigen, remember they are naive. When they are naive, they have a certain type of B cell receptor on their surface. Okay, so B cells, they have B cell receptors. Those are very, very specific, right? They have B cell receptor on their surface, and when they are naive, they have actually 2 types of B cell receptors on their surface. So here is your B cell. They are going to have one receptor that is an antibody. The B cell receptor is antibody. Okay, when it's bound to the cell surface, we call that the B cell receptor. When it's just floating around, not bound to the surface, you just call it antibody. Okay, so the naive B2 type, it's going to have this B cell receptor that is called IgM. So IgM, when it's bound to the surface of the cell, is only a single unit there. Is not a multimer. It's a monomer. Okay, and the cell is also going to have this other antibody that is also B cell receptor that is called IgD. Okay, so these B cells, they are mainly going to have IgM, but they're also going to have some IgD on the surface. Okay, and that's your naive B2 cell. After they see antigen, after they get activated, they will start to change their receptors. They're going to start changing the antibody. So they're going to undergo this process called class switching. So class switching really has to do with the type of antibody that the B cell produces. Okay, after it sees antigen, after it gets activated. Okay, so after your B cell becomes an effector cell, it is going to be able to interact with T cells and is going to further differentiate and become even more specialized and more effective at killing pathogens. Okay. All right. So here and then here, any questions about what's happening here? Everybody cool so far? Yeah.
Do they change their DNA?
Yes, they do change the DNA. That's a good question. Yeah. Yeah. When you get to the cell's development, we're going to see how that happens. Okay. All right. So here, with their natural killer cell, these guys do not change their DNA. Okay, they have what they have. They also do not have a T cell receptor. So natural killer cells, they're kind of cousins of the T cells, but they do not have the antigen T cell receptor. Okay, what they have is this receptor for this type of cytokine that is called interleukin-2. That's a very important cytokine, as we're going to see later. Okay, so interleukin-2 receptor is on the surface of those cells. And since these cells do not have an antigen receptor, they are not going to detect antigens. Okay? So what they're going to do, in comparison to the other T cells that have to detect antigen presented by MHC molecules, right? So these guys, what they're going to do, they're going to detect the absence of MHC molecules on cells. Okay, this is a very, very important mechanism of how natural killer cells do their killing. Okay, so here let's draw a regular cell. So this is a regular cell. It has a nucleus over here. So this is a nucleated eukaryotic cell, right? That's kind of redundant. So this is an eukaryotic cell. And where do you find eukaryotic cells in your body? This is when I check if you're awake. Everywhere. Okay, all of your cells except for your, well, actually all of your cells. Exactly, except for your microbiome. All of your cells are eukaryotic cells. Okay, except for your bugs, of course. I mean, bacteria. Okay. All right, so all of your cells that have a nucleus, they express one type of MHC molecule, and that is MHC type 1. All nucleated cells in your body, okay? Oh, okay, I just messed up my previous sentence. Which ones are the cells that do not have a nucleus and you have plenty of them in your body and they're not part of the microbiome?
Red blood cells.
Red blood cells, yes. Okay, so here you have your red blood cell. Okay, red blood cells do not have nuclei, right? So do red blood cells express MHC1? No, they do not express MHC1. Okay, all right. All the other cells in your body express MHC1. So everything is cool. Now let's say that this body cell got infected. So now you have a little virus. I put kind of like virus here, okay? Kind of like upset virus inside of the cell. Got infected, okay? A cell that is infected, first thing that it does, it is going to slow down the production of that molecule, of MHC1, okay? So you are not going to see MHC1 on the surface. You're not going to see a lot of MHC1 on the surface of the cells that are infected. Now comes this guy, the natural killer cell. It goes over there, it's patrolling everywhere, and it's looking for eukaryotic cells that have MHC1, right? So here's the job of natural killer cell. Goes around, and how do the cells recognize things? They don't have eyes. They have to go by feeling, right? They go with sensors. They go around and they go, okay, eukaryotic cell. You have MHC1, you're good. Eukaryotic cell, okay. You don't have MHC1, something is wrong with you. I'm just gonna kill you just in case. They're kind of like, doesn't want to ask a lot of questions. You know, it's like, you don't have MHC1, that is a signal for me to kill you. Do you have other molecules that are telling me not to kill you? It's like, no, So you're dead. Pretty much it. Okay, so this is one important way that your natural killer cells go around killing cells that do not have MHC1. Yeah.
What happens if it detects MHC2?
It doesn't detect MHC2 because
Yeah, but like if it has it and it doesn't have MHC1.
a cell that has MHC2 also has MHC1. Yeah. All nucleated cells have MHC1. And only very specialized antigen-presenting cells will have MHC2. Okay, as we'll see next. Yeah. But then your natural killer cells are going to look for presence of MHC1. Okay, I'm going to see why also later. Okay, so no MHC1 is going to kill the cell. And usually it will kill the cell by telling the cell to die. It will go by apoptosis, and this is the next slide here. Okay. So innate lymphoid cells, what are they doing? So this is your innate lymphoid cell right there. So it did not detect MHC1, and this cell here actually has a lot of viruses growing over here, right? So the cell interacts with the target cell and is going to tell the target cell to commit suicide. The pathway, as we're also going to see later in more details, that the lymphoid innate cell is going to tell is going to be by this signaling pathway called Fas and Fas ligand pathway. Right, so it's going to interact with the Fas receptor and is going to tell the cell to die. Okay. Besides the Fas and Fas ligand interaction, the lymphoid innate cell can also dump granules. You see those little things over here? Those are granules packed with deadly stuff that is called granzyme and perforin type of molecules. What they do, they punch holes on the surface of that cell. If you punch holes over there, then parts of the cytoplasm of the cell are going to spill out, like this. So the cell can come out. So the cell is going to die. Okay? So two ways of killing the cell. Either you tell the cell to commit suicide by apoptosis. So that's your Fas and Fas ligand pathway. So this is apoptosis. Or you punch holes on the surface of the cell and you allow the cell to die anyway. Right. Okay. Right. So here. So overview here of your adaptive cells function. Okay. So adaptive, that means natural killer cells are not part of this, right? Those are innate. All right, so adaptive, remember you have your helper T cells. What they do, they're going to help to activate other cells, okay? Activate what? They're going to activate certain innate cells, and they're also going to activate the cytotoxic T cells, and they're going to help the B cells to differentiate properly, okay? So those are helper cells. Then you have here your regulatory T cells. Remember, that one suppresses every cell that they come across. They secrete some suppressor cytokines, okay? Then you have here your killer cells, your cytotoxic, right? Cytotoxic T cells. So these guys, they are going to recognize things that are part of intracellular pathogens that are present in some other cells, like infected cells. All right, so these guys can kill infected cells by recognizing non-self presented by those cells. All right. And then you have your B cells here, right? Remember your B cells, mainly you're going to work with, or you're going to look at your B2 type. And you call them B2 before they see antigen. After they see antigen, they transform or they mature into plasma cells. And those are the ones that are going to give you your antibodies. Okay, so you look in the bone marrow here. So here's your bone. Okay, so here's the marrow. you are going to see inside the bone marrow, you're going to see a lot of blood vessels. And then you're going to see here some of your hematopoietic stem cells. Those are not differentiated, right? And you're going to see other types of cells. You're going to see the stromal cells that should be here somewhere. Okay, I don't see them labeled here. Stromal cells should be around here, okay? Besides stromal cells, you also have helping cells. I think, yeah, you have macrophages, you have some other types of cells that are present there to start teaching your hematopoietic stem cell how to differentiate. So all this process of differentiation here is going to happen inside of your red bone marrow. Then after your B cell is mature, then it's going to go into the blood vessel and it's going to travel to your lymph nodes. Okay? So everything is happening in the bone marrow. After it's mature, it's going to come out, which means it's going to migrate out of the bone marrow, and then it's going to reach your lymph nodes, or spleen sometimes, mostly lymph nodes. Okay. Oh, here is another picture that is interesting. Okay. So that's the story with the B cells. With the T cells, you have two primary organs that allow development of your T cells, right? So T cells, they start in the bone marrow, then they're going to travel to the thymus, right there, right here, right? They're going to travel to the thymus and they're going to finish their development there in the thymus. Okay, so in the thymus you have two important regions. You have the region that is close to the edge of the thymus, you call that the cortex, and then you have a region that is close to the center, and you call that the medulla. Okay, so you have like cortex on the outside of different sections. So cortex on the outside, medulla on the inside. So what happens here when the developing T cell, which call that a thymocyte, gets to the thymus, it starts going to the cortex, and it starts to be exposed to all different types of testing of the receptor. Okay, so two important things that you have to know here. The first thing that happens when the cell gets to the cortex is that it is going to be tested whether it has useful T cell receptors. Okay, so if this cell has useful T cell receptors, then the cell is going to be allowed to survive. Okay, that is what you call your positive selection. Okay, so positive selection is useful T cell receptors. Okay, useful receptors. Okay, positive selection. Now, if it does not have useful receptors, it's going to die. Okay, so now let's say that your cells have useful T cell receptors, and now it migrates, it goes from the cortex towards the medulla. When it gets to the medullary area, it is going to undergo another type of selection. Okay, so now you're going to check whether your T cell receptor attacks self stuff. Okay, so if this T cell receptor is working but is attacking your own proteins, then you have to get rid of the cell. That is what you call your negative selection. Okay? If your T cell receptor attacks self molecules, you have to kill them. All right? So if attacks self, then you kill them. That is called negative selection. Everybody cool? Positive and negative selection? Yeah, am I going to ask you about that? Yeah. Okay. Yeah, so for the negative selection, if your T cell receptor attacks self-antigens. Okay? If it's attacking self-antigens, you have to kill it. So that is your negative selection. So then it's like that, kill it. Okay? Sometimes things go wrong during these processes. Okay, so things can go wrong in the medulla, and then you do not really detect a cell that is attacking self-tissue, and then the cell is allowed to fully mature and come out of the thymus. Then you're going to have a problem. You're going to have autoimmune disease. Right? That's a big problem. All right. That's one of the ways you can get autoimmune disease. Okay. Okay, so now that we looked at major characteristics of all the cells involved in the immune system, right? We look at innate and adaptive. So now we're going to start looking at the big picture and how all these things are going to start working in more details, okay? So here looking at the entire lymphatic system, right? So what is the lymphatic system really? Well, it's basically this system that is always monitoring your fluids and monitoring the tissues and really making sure that pathogens do not cause disease, right? So what makes up here your lymphatic system? You have a lot of tubes, you have a lot of pipes, and you have centers that are meeting places, right? Besides all that, you also have the organs that produce those immune cells, right? And basically the fluid that is going around those specialized lymphatic pipes, you call that fluid lymph. Okay, so here is the idea of how you get the lymph, right? So basically you have blood going through your arteries, and once they come out of the arteries and they go through the capillaries, you have some leakage of some fluid out of the capillaries that get into the interstitial space. So that is outside of your capillaries, outside of your arteries and veins. Okay, so the fluid that leaks out, it bathes the tissues, it takes nutrient to the tissues, but then it cannot just be stuck in the tissues. You have to reabsorb that fluid somehow back into your circulation. So this is where your lymphatic vessels are really, really important. So those lymphatic vessels are strategically placed close to those capillaries, close to the capillary beds. And they are like open straws. They are open-ended type of vessels. Okay? So what happens here is that when the fluid comes out, it will interact here with this little lymphatic capillary, and it's going to be brought inside the lymphatic capillary just by capillary action. Okay? So it will be absorbed here in the lymphatic capillary and is going to be pushed up the lymphatics, and during this process the fluid is going to pass through nodes, through those lymphatic nodes, and the lymph is going to be filtered like that. So the liquid that gets back to your lymphatic vessels, you call that liquid, you call it lymph. Okay? So when it just comes out of the blood vessels here, or the capillaries, you call that interstitial fluid. So when it gets to the lymphatic capillaries, you call that lymph. Okay? All right. So what makes the lymphatic system? You have your primary lymphoid organs. Remember, that's the bone marrow and the thymus. And then you have all these pipes network, the conducting network. You have the very, very tiny ones. There are the capillaries. Those do the capillary action to suck the liquid back into the vessels. Right? Then you're going to have the bigger lymphatic vessels. Inside these lymphatic vessels, you're going to find one-way valves to avoid liquid from leaking back out. Okay. Then the vessels, they're going to join bigger and bigger vessels, and when they get to the very big vessels close to your neck, you call those lymphatic ducts. So you have two major lymphatic ducts here. You have the right subclavian, the duct that dumps into the right subclavian vein. Okay, so you have the right lymphatic duct right there, the green one right here. Okay, and this right subclavian, the right lymphatic duct, it really brings the liquid from the upper part of the right upper part of the body. Okay, so this part here, the right arm, this right upper part of the body gets dumped into the right lymphatic duct. And then the lymphatics that get up here and get together to this big thoracic duct that is on the left side. Okay, the lymphatics that drain to the thoracic duct that dump into this left subclavian vein here, they bring fluid from the rest of the body. Okay, so here for the left, they bring fluid from like all the rest of the body. Okay, that goes into your left subclavian vein right there. Okay. And here I put a video link for these amoeba sisters. Actually, one of you mentioned last time about these amoeba sisters, so I checked it out, and it's actually really cute. I like them. So nice 8 minutes video for that. Okay. So more components of your lymphatic system, right? You have your secondary lymphoid organs, right? We have briefly mentioned before. So here you see them in more details. You have the lymph nodes all over the body. How many of them? A lot of them. 600 to 700 of those little lymph nodes. You have certain areas that they have more of a concentration of those lymph nodes. Okay, but mainly they are distributed all over the body. You have here around the close to the leg. Okay, you have close to the armpit, and you also have close to the neck. Those very, very important areas for concentration of lymph nodes. What is it that the lymph node is doing? Is filtering that lymph. That lymph is coming from the interstitial fluid, right? That leaked out of the blood. So lymph nodes are really filtering everything that is not in the blood, that leaked out of the blood. Okay? Important function for lymph nodes. Besides that, you also have the spleen right here. Now, the spleen is very vascular. It really filters the blood. So whatever is going through the blood circulation is going to go through the spleen, and if you have some pathogens that are in the blood, those are going to get filtered in the spleen. Okay, the spleen also has other functions, but it's very, very important for filtering pathogens from the blood. Right? So lymph node filters the lymph, the spleen filters the blood. And then you have those tonsils and adenoids. Those are mainly glands that are in the back of your nose, in the back of your mouth, right? And those trap microbes that you might inhale or smell or swallow. So a lot of the microbes get trapped by the those secondary tissues, tonsils and adenoids. And then you're going to have here your mouth, right? So that is your mucosa-associated lymphoid tissue, and you're going to find that in connection with your mucosal tissue, okay? Mainly, you're going to find that in the intestines. Okay, so you have the small intestine here, a lot of those mouths. When you look at the mouths from the intestine, the most prominent ones are the Peyer's patches. Okay, so those are in the intestines here. All right. All right. So here are some interesting clinical correlations on problems that can happen with the lymphatic system in general. Right. The first problem that you see here is lymphedema. Have you heard of lymphedema? Yeah. Main causes of lymphedema are secondary causes. Okay. So that can mean that if somebody has cancer, you can have a problem of the lymphatics or occlusion of the lymph nodes. So all the part that is supposed to be drained through the lymph node, all the rest of the part gets accumulation of fluids, right? If you have a certain type of cancer that is occluding the lymph node. One known cause of lymphedema is actually a parasite, a parasite that causes infection through these lymph vessels, right? The name of this parasite is what? Filariasis. And another name for this lymphedema is called elephantiasis. Yeah, elephantiasis. And the way that you see that you can identify this is, like for example, if you look at the picture here, and if you have the lymph nodes of one leg that are clogged up, so then the leg starts to accumulate this fluid, right? So this is just edema, very large edema. And you can recognize lymphedema basically by the asymmetry of the limbs. Right, so you see one limb that is totally out of proportion in comparison to the other. So that's the lymphedema. Okay. Yeah.
So that means that the fluid just stayed in the interstitial space.
Fluid stays there. It has nowhere to go, so the tissue starts to expand. So yeah, to accommodate all the fluid that gets stuck in there. Yeah. All right. So
and
this.
That cause like damage to like the blood vessels.
It causes a lot of damage. Yeah, yeah. Because you can imagine with all this pressure, the circulation can't really do a good job. Yeah, it's just putting too much pressure. Yeah.
So it could cause like blood clots.
It can cause all types of problems. Yeah. And I don't really know how they take care of this. I think mainly surgery. In case of the parasite, there might be some drugs. Yeah. I didn't look up the cures. There must be some cure if the infection is caused by the parasite. There must be some drugs to kill the parasite. But in other cases, like in case you had a cancer and had to have surgery and remove some of the lymph nodes, then it has to be a bit more difficult. Usually the person will use compression garment and massage and try to do lymphatic drainage to alleviate the symptoms. Yeah. And sorry, you have a question? It was the same question. Okay. All right. Now, this comparison here to another problem that is not well known but is getting more and more attention lately is lipedema. Lipedema is not lymphedema. Okay. Lipedema is another type of big problem. Okay. This is a chronic inflammation and chronic disease of fat tissue, but not all fat tissue, especially fat tissue related to from the hips down, mainly the legs. Okay. So basically, until now, you say no, lipedema, the disease they call fat. So you see somebody with a very huge, waist down, bottom legs, and you go like, oh, it's just fat. And yeah, in case you need a visual effect, I have lipedema everywhere. Okay, from the waist down. I have been fighting with it since adolescence. Usually that's when it shows up. If you have a Latin background or African American background, most likely you're going to have some lipedema genes. The person that is really curvy, yeah, lipedema. Okay, so that's like over 10% of the women population. Yeah, it's a women problem. It's a hormonal problem. It's a fat storage problem. It's the kind of fat that you can exercise as much as you want and the fat will not go away. I tell you, it doesn't go away ever. Unless you go through surgery, and they do have specialized surgeries for lipedema now. Yeah.
So do you develop lipedema later, like when you're pregnant?
Yes, you can develop, if you have a tendency for it, you can develop it during major hormonal changes. Adolescence, pregnancy, and menopause. Yay. And then what do? Oh, there are several things that you can do, right? None of them are really effective. I tell you, well, just the fat in this lipedema tissue, the fat absorbs the nutrients from your blood. So you can be starving, literally, you can be starving, and the fat is still growing. It sucks up the nutrients that you eat. Isn't that frustrating? Very, very frustrating. Actually, in 2019, I decided I didn't know about the lipedema. These studies, the notion of lipedema is only coming up in the last 5 years or so, okay? So by 2019, I didn't know much about it, and I said, you know what, I'm going to get rid of this fat. So I decided to walk the Camino de Santiago. You guys heard Camino de Santiago? More or less. There are many Caminos in Europe, so you can have the Camino from Italy, from France, and it goes across Spain, and you get to Santiago de Compostela. It's a long walk, like 500 plus miles of walk. So I took about 30 days, and I did 15 days hiking, about 260 kilometers, and then I rented a bike, and I did the rest of biking, and I tell you, it's only uphill. They have the mountains, and they never go down. It's all uphill. Most of them. Anyway, the downhill is fun too. But I did it all, and then what I see all my fellow Camino family. Everybody's losing a lot of weight, and they are eating like crazy. You're like exercising 8 to 12 hours a day, and you can eat a lot of these carbs. And those people are losing weight, and they're like shrinking, and I'm like, nothing is happening. And then we finished the Camino. I managed to get rid of 3 pounds. I think it was just water weight. The rest of the people got rid of 20 to 25 pounds. Tell me about frustration. Yeah. And then after that, I said, oh, that's it. I gotta, make peace with the legs. At least they work, But, and then I started finding out about the lipedema. It is a problem, So if you want to learn more about it, there are several lipedema sites that you can really find out more about that. So interesting thing about the immune system, right? You thought it was all about memorizing cells and the receptors and development of those cells. No, we have actually real life problems, right? Okay, so let's get to the more details on the pathways and stuff. Right. So here we start with our nitty-gritty details on your innate immunity, right? Specific aims of this. You have to know, which I already did before, the characteristics of innate immune response. And now we're going to look more into those pattern recognition receptors, where they are, what they are doing, right? The importance of them. We're going to see that you have 3 main types of PRRs. Some of them are on the surface of cells. Some of them are inside the cells. And some of them are just floating around, patrolling the fluids, right? So all different types of PRRs. Once you activate a PRR, this is the main mode of operation of the innate system cells. Once you activate that, then you are going to activate certain types of inflammation, right? So we're going to look at an NF-kappa B type of inflammatory process. Okay. Besides NF-kappa B, we're going to look at another type of inflammation and cell death that is mediated by NLRP3 cell signaling. Okay, so this is also pathway of inflammation, and actually this pathway of NLRP3 happens through this process that is involving what you call the inflammasome type of mechanism. Okay. So here, then just to reiterate here, what is your main response of innate immunity? It's very, very fast, and it does not hold memory of the thing that it has seen before, right? So things that are important for that innate response, you're going to have certain enzymes, as we're going to see in more details, which enzymes and what they're doing, right? You're going to have enzymes that are present on your skin, present in your guts, and present in respiratory system. Okay. Besides all of those physical barriers that we have seen already, right? Parts of the physical barrier that I had not mentioned before is really like cilia. So when you look at the airways, for example, you see a lot of this cilia. Well, not really in the nose, although you see like hair in the nose, right? But that's the hair. Like when you go deeper into your respiratory system, you see the cilia. So cilia is very important for trapping particles of either pollution or pathogens. And you have this type of ciliary escalator that brings it up to the mouth or the nose, and then you kind of, you spit them out. Okay, so cilia is important to, as a physical barrier. Okay, so what are we looking at here? So this is that picture that is showing you the entire immune system, showing you physical barrier and innate immunity, and then connection with adaptive immunity. So we are looking here at this area right here, the beginning of the immune response. Okay, so something has to cross the physical barrier and is going to be attacked by those innate cells and is going to be processed in this area right here. And you know the innate cells are usually very, very efficient because they eliminate, whatever comes across, and we never even find out about it, right? We don't even know how efficient they are because the only time we find out about a breach in the system is when you get sick. But if you are getting sick, that means this system was not sufficient to eliminate the pathogen, and you had to activate your adaptive response. And then you remain sick until your adaptive response really kicks in. Okay. So here, you have a lot of antimicrobial proteins and peptides that are a part of your innate system. Okay, so you call them antimicrobial proteins, or AMPs. Okay. Okay, so you're going to find them on the skin, epithelial layers, okay? And important proteins that you're going to find here are lysozymes. Okay. So lysozyme is very, very important against bacteria. So this lysozyme is going to break some sugar bonds, some disaccharides bonds. So you have here NAM and NAG sugars that are typical that on the surface of certain bacteria, right? And especially gram-positive bacteria, right? So this lysozyme goes there and breaks these sugars, and that is, if the sugars, since they are part of the cell wall of the bacteria, it ends up spilling the guts of the bacteria, and the bacteria will die. Okay, so lysozyme is very, very important part for that. Besides lysozyme, you have a lot more of these peptides. Like how many? Oh, look at this, about 22,000 of them. A lot of them, right? They're really everywhere in your body, and especially with, of course, in contact with the outside world. All right. So how is it that they work? Well, first, where are you going to find them? You're going to find it on the skin. You're going to find coating the intestines, right? Coating the mucus. You're going to find in breast milk, also saliva and tears. So anything that is in contact with the outside environment, you're going to find those peptides, antimicrobial peptides. The strategies of those peptides. One strategy that I already mentioned is breaking the sugar bonds on the surface of bacterial cells, also surface of fungi. Besides that, these peptides, they can also hold on to iron molecules. And if they gobble up the iron, certain bacteria cannot grow. So sequestering iron is a very good strategy to slow down the growth of bacteria. Okay? Besides breaking the cell wall, sequestering iron, they can also mess up directly the bacterial membranes. Okay? Besides all that, they can bind to the surface of bacteria, and here's a little bacteria, right? And here's the surface of bacteria. They can interact with those peptides, and that will block the interaction of these bacteria to certain target tissues. So the bacteria, it will not be able to interact with the site of infection that would be infecting. Okay. So besides that, you also have antifungal action and antiviral action of those molecules, of those proteins. Okay. All right. Where are these proteins coming from? Well, either you produce them or your friendly bacteria can produce them for you. So this is one job of your commensal bacteria, right? So bacteria in your intestines and on the surface of your skin, they can produce certain types of these peptides, and we know a very few famous ones like penicillin, okay, vancomycin. Those are the two that most people have heard of. And then you have this other one, polymyxin B. Those are all produced by your friendly bacteria to kill the bad guys. Okay? Right? Besides killing the bad guys, those peptides have other functions, like for example, they help out with reconstituting the tissue and communicating between cells. Okay, cell communication and wound healing is very, very important feature of those peptides. All right, so some peptides that are good to know, right? So here we go. Yeah, there are something right here, but mainly they have, functions that are easy to remember. So we mentioned lysozyme. You know where to find them, right? Mucosal secretions. And what is it does? Is doing is really breaking down the cell walls of bacteria. And then you have this guy here, lactoferrin, ends up in ferrin, has to do with iron. This is an iron sequester, right? Then you have some protease inhibitors. Okay? They are going to slow down, or is going to break down certain proteins, not really the cell wall, but it's going to break down certain proteins of your pathogens. Besides all that, you also have these guys here, surfactants. Okay, surfactant protein. You're going to mainly find it in airways, okay, like in the lungs. Okay. And what they do, they are going to block the interaction of the pathogen with the tissue that it wants to infect. So it coats the pathogen and tags it for destruction. Right? So these surfactants, they actually can work as opsonins. That means coat the pathogen and make it really tasty for phagocytosis. So some macrophage is going to go there and eat it up. Okay. And then you have this other type of protein here, ApoL3, that is important against gram-negative bacteria. Remember I mentioned here your lysozyme for gram-positive and this ApoL3 for gram-negative. Okay. All right. And you have more of them, defensins. Defensins are going to disrupt membrane, okay, bacterial membrane. And this guy here, look, he's statins. Okay, that's very important in the saliva. Okay, that is important for killing or breaking down fungi. Okay, fungi, especially for fungal cell walls. Okay, and well, dermicidin, this one is really on the skin, and that's since it's derm to it, so like dermatological, it's easy to remember it is important for antibacterial function and antiviral function and antifungal function on the skin. So how is it that the thing is happening? How is it that your innate system is really putting all the stuff to work? First thing it has to do, it has to recognize there is a problem. Once it recognizes there is a problem, and this is with your PRRs, it has to activate some response. In this activation, there is internal activation of the cell, and there is some external activation because the cell is going to dump out products like cytokines and inflammatory proteins to cause some type of inflammation. Inflammation is a very, very important feature of your innate cells, innate immune system. Okay, so it's going to produce acute phase proteins. Those are your inflammatory proteins, inflammatory cytokines. Okay. Besides that, you have the system that is also responding by increasing surface receptors. So they want to make cells sticky in order to interact with other immune cells. So by making the cells sticky and by putting cytokines out in the environment, they are going to bring other cells to the site of inflammation. That gives you the recruitment of other cells, right? After everybody's over there joining the party, they are gonna kill the bad guy, okay? After the bad guy is gone, these people really have nothing else to do, so they are going to slow down and they're going to start, dying, okay? Without the stimulus, they start dying. So that's your reduction. While they are dying, you also have tissue repair that is happening. All right. So here, yeah, I have a lot of words for you to read here, but the most important thing here is that your innate immune system a lot of times is not sufficient, right? You have to activate your adaptive immune system. The reason why your innate immune system is not very sufficient to just get rid of all the pathogens is because pathogens are very clever. Pathogens start to develop certain types of strategies to go around all of these defense mechanisms that the innate immune system has. So the pathogens develop resistance to innate system strategies. Okay, the resistance that they develop, that is really what you call virulence of those pathogens. The more resistant they are, the higher their virulence. Okay. Okay. Oh, finally, we got a question here. So here, okay. So this is asking which of the following is representing the one adaptive immune response. So now you got to select the one that is adaptive. Okay, so everybody answered? Okay, what is your answer? It is D. Adaptive, you think B cells and T cells. Everything else was innate. So here, let's look more into those myeloid cells, right? So myeloid is mainly your innate. They are really going to try to destroy the pathogen and keep everything quiet, right? And you know what those cells are. And then remember, you have also innate cells that are your natural killer cells, okay? And you have your innate lymphoid cells. Okay, those are all part of your innate system. And here's when the fun starts, right? So your innate lymphoid cells, you actually have 3 different types of those innate lymphoid cells. Those are your cousins of your T cells, right? Each one of these types is going to be activated depending on the type of pathogen that you are fighting. Okay, so the first type of innate lymphoid cell that you have is the type 1. Okay, and you're going to fight here mainly intracellular bacterial infections. Okay, type 1 here, you see in this diagram here, is going to be mainly intracellular infections and also some types of viral infection. And as you're going to see with a lot of mechanisms of innate defense, you have several pathways that are redundant, You have one pathway that fights virus and bacteria, another pathway also fights virus and bacteria. Well, the reason is that if one pathway fails, then the other pathway can pick up the function and you still survive. Okay? So when you look at type 1 response in innate cells, and you're looking here at type 1 innate lymphoid cell response, you're mainly looking at intracellular bacterial pathogen. Okay, so an important characteristic of this cell is production of interferons. Okay, interferons in general, they are the first line of defense against viruses. Okay, and of course viruses are intracellular pathogens. Okay. So bacterial, intracellular bacterial infections, and also some viral infection. Okay. All right. So besides your type 1 lymphoid cell, you have a type 2 lymphoid cell, and this is the type that is very important against fighting parasites like helminths, so worm types of infections. Right? And one interesting thing about these cells is that I probably mentioned before that in a more advanced type of social setting in which you are not really exposed to a lot of parasitic worm infections, those cells get quite bored because they don't really have a job to do, right? So they start looking for things to do, and then they find something like attacking things that are inoffensive. They find it fun, and this is when you get allergic reactions, right? So type 2 immune responses really have to do with fighting parasites or development of allergies, okay, hypersensitivity type of reactions, okay, type 2 cells. Then you have your type 3 response, or type 3 cells. Those are really more important for fungal infections, and also for some viral infections, but mainly it's going to fight extracellular type of pathogens, okay? And what this is going to do is mainly going to cause inflammation. Those type 3 cells are very, very good at causing inflammation. They will dump a lot of inflammatory cytokines, and they're really going to ramp up that inflammatory process. Okay. So that goes hand in hand with the types of immune responses that you have to develop depending on the type of pathogen that you face, right? So the type 1 immune response has to do with intracellular bacteria and viral infections. I should put there too, okay? Type 2 has to do with those parasitic worms and also diseases like hypersensitivity, right? Allergies. And then type 3 really have to do with inflammatory type of responses, because you're going to fight extracellular type of pathogens. When type 3 responses go crazy, you can also develop long-term chronic inflammatory problems, okay? Once you trigger chronic inflammation, there's no way to reverse that situation and go down to no inflammation. So it's very important to avoid chronic inflammation and really keep healthy immune system the longest time possible. Because by the time you get to be 50, 60, 70, and if you develop chronic inflammation, then it's like it's not good. It just gets worse. Okay. All right. So here, this is important. It's in red, it's bold, it's underlined. So you gotta know this, right? 2 major types of protective reactions of your innate immune system. One of them is inflammation. The other one is antiviral defense. Okay, inflammation and antiviral defense. How are you going to get inflammation? You're going to dump proteins in the environment. There are cytokines, and they are pro-inflammatory cytokines. You're also going to interact with plasma proteins that are produced by the liver, right? So that is important for development of inflammation. There are 3 cytokines that you absolutely have to know, so let's just get introduced to them now. 3 pro-inflammatory cytokines: interleukin 1, interleukin 6, and tumor necrosis factor. Those are very, very important inflammatory cytokines. Okay. All right. Okay, so that you also have your antiviral defense here, especially the production of interferon type 1. Okay. Okay, this is just for your review. Okay. So here we're finally going to get into your nitty-gritty of pattern recognition receptors. Okay. Okay, so let's see what's going on here. You have all different types of PRRs. So you have PRRs on the surface of cells. You have them inside vesicles. You have them floating around in the cytoplasm. You have them on the surface of mitochondria. Okay, so they are really, really everywhere. And they are specialized on detecting patterns that are from pathogens and some damage-associated patterns as well, okay? So important ones that you have to know. Toll-like receptors. You're going to find these guys here that they look like a little hook, like this. And usually they will be on the surface of cells. They'll go through membrane, okay? So here's an example of toll-like receptors, okay? They are very important here for detecting all sorts of things. They can detect viruses, bacteria, and fungi, all sorts of things. And once they are activated, they induce phagocytosis of whatever they interacted with. Okay, so they induce phagocytosis and further intracellular responses to eliminate that pathogen. You have other ones called C-type lectin receptor. This is also on the surface, although it's not showing here. And it can detect specific bacterial components. You get these other guys here, Rig-I receptor. Those are in the cytoplasm. Where are they? Okay, it's too much information here. Rig-I, they are important for interacting with foreign RNA and DNA, mainly viral RNA type. Okay. And you have these other types here, nod receptors. Those are important for also intracellular detection. And yeah, we do have more specifics going around. But basically they are everywhere, on the surface, in the cytoplasm, floating around. We have better slides for that. Okay, so here we go. Basically we know those receptors are produced from genes that are fixed in the DNA. And you have about hundreds of them, not a lot, but those, okay, so here about 100 different types of receptors, okay, and those can detect about 1,000 or so different patterns, okay, so the very limited detection system here. Now, remember, those are encoded in the genes, and they do not undergo any form of genetic modification, right? When you compare that to receptors of your adaptive immune system, and look at these numbers. The adaptive immune system receptors are your T cell receptors and your B cell receptors. Look at this detection of antigen, 10 to the 7 possible detections of antigens. Like, how much is that? A lot, right? I mean, just like 10 to the 7. 1, 2, 3, 4, 5, 6, 7. That many, kind of this many, okay? A lot. And your adaptive immune system receptors, those are the ones that undergo genetic recombination. Okay, so you have basically infinite number of receptors that you can produce. All right, so here are some important PAMs, important PRRs that you should know. Okay, I'm going to stop after this slide here. Okay, what is it the PRR can detect? It can detect nucleic acids that are not yours. All right, so CpG islands, double-stranded RNA, that's an important one for viruses. Okay, those can detect proteins also they are not yours, like flagellin in bacteria. Okay. They can detect cell wall stuff, like LPS in case of gram-negative bacteria, lipoteichoic acid in case of gram-positive bacteria. They can also detect carbohydrates, like mannan for fungi, glucans for fungi. So go over this is important for the detection. Yes, I will stop here and we'll pick up from this slide on.